Hi everyone, today we are going to talk about lipid. So as we all know, like an HIV particle exists an infected cell by membrane budding. So cellular membranes are highly dynamic structures that spontaneously self-assemble. Biological membranes serve several additional functions indispensable for life, such as energy storage and information transduction that are dedicated by the proteins associated with them. In this chapter, we will examine the properties of membrane proteins that enable them to exist in the hydrophobic environment of the membrane while connecting to hydrophilic environment. The boundaries of all cells are defined by biological membranes, dynamic structures in which proteins float in a sea of lipids. In addition to an external cell membrane, we call it the plasma membrane. Eukaryotic cells also contain internal membranes that form the boundaries of organelles, such as mitochondria, chloroplasts, peroxisomes, and lysosomes. External and internal membranes share essential properties. These figures are the subject of this chapter. The hydrophobic properties of lipids are essential to their ability to form membranes. Most lipids owe their hydrophobic properties to one component, their fatty acids. Fatty acids are long hydrocarbon chains of various lengths and degrees of unsaturation that terminate with carboxylic acid groups. The systematic name for fatty acid is derived from the name of its parent hydrocarbon by the substitution of OIC for the final letter E. For example, the carbon-18 saturated fatty acid is called octadecanoic acid, so ended with OIC, because their parent hydrocarbon is octadecane, so end with E. Carbon-18 fatty acid with one double bond is called octadecanoic acid. So with two double bonds, we call this diionic acid, and with three double bonds, we call it like a trionic acid. So etc. The structures of the ionized forms of two common fatty acids, palmitic acid and oleic acid, so as shown in this slide. Fatty acids in biological systems usually contain an even number of carbon atoms, typically between 14 and 24. So the 16 and 18 carbon fatty acids are most common. The hydrocarbon chain is almost invariably unbranched in animal fatty acids. The properties of fatty acids and of lipids derived from them are markedly dependent on chain length and the degree of saturation. Unsaturated fatty acids have lower melting points than do saturated fatty acids of the same length. By definition, lipids are water-insoluble biomolecules that are highly soluble in organic solvents, such as chloroform. Lipids have a variety of biological roles. They serve as fuel molecules, highly concentrated energy stores, signal molecules, and messengers in signal transduction pathways, and components of membranes. Here, our focus is on lipids as membrane constituents. The three major kinds of membrane lipids are phospholipids, glycolipids, and cholesterol. Phospholipids are abundant in all biological membranes. A phospholipid molecule is constructed from four components. one or more fatty acids, a plant form to which the fatty acids are attached, a phosphate, and an alcohol attached to the phosphate. The plant form on which phospholipids are built may be glycerol, a 3-carbon alcohol, or sphingosine, a more complex alcohol. So phospholipids derived from glycerol are called phosphoglycerides. In phosphoglycerides, the hydroxy groups at carbon 1 and carbon 2 of glycerol are esterified to the carboxy groups of the 2 fatty acid chains. The carbon 3 hydroxy group of the glycerol backbone is esterified to the phosphoric acid. When no further additions are made, the resulting compound is phosphoattitude. So the simplest phosphoglyceride. The major phosphoglycerides are derived from phosphoattitude by the formation of an ester bond between the phosphate group of a phosphoattitude and the hydroxy group of one of several alcohols. The common alcohol motifs of phosphoglycerides are the amino acid serine, ethanolamine, and chlorine, and glycerol, and inositol. So the structural formulas of phosphoattoglycerol, and other principal very important like phosphoglycerides, namely phosphoattidyl anodamine, and some other like fatty acid serine. These all are given in this slide. So sphingomyelin is a phospholipid found in membranes that is not derived from glycerol. Instead, the backbone in phenolglycine is sphenol. Instead, the backbone is sphenoxane, an amino alcohol that contains a long unsaturated hydrocarbon chain. In sphingomyelin, the amino group of the sphenoxane backbone is linked to a fatty acid by an amide bond. In addition, the primary hydroxy group of sphingosine is esterified to phosphoric chlorine. The second major class of membrane lipids, glycolipids, are sugar-containing lipids. Glycolipids differ from sphingomyelin in the identity of the unit that is linked to the primary hydroxy group of the sphenoxane backbone. In glycolipids, one or more sugars are attached to this group. The simplest glycolipid, called a cerebroside, contains a single sugar residue, either glucose or galactose. So more complex glycolipids, such as gangliosides, contain a branched chain of as many as 7 sugar residues. Cholesterol, the third major type of membrane lipid, has a structure that is quite different from that of phospholipids. It is a steroid, built from 4 linked hydrocarbon rings. A hydrocarbon tail is linked to the steroid at one end, and a hydroxy group is attached at the other end. So cholesterol is absent from prokaryotes, but is found to varying degrees in virtually all animal membranes. It consists almost 25% of the membrane lipids in certain nerve cells, but is essentially absent from some intracellular membranes. The membranes of the archaea differ in composition from those of eukaryotes or bacteria in 3 important ways. 2 of these differences clearly related to the hostile living conditions of many archaea. So first, the non-polar chains are joined to a glycerol backbone by either rather than ester linkages. Second, the alkali chains are branched rather than linear. They are built up from repeats of fully saturated 5-carbon fragments. These branched, saturated hydrocarbons are more resistant to oxidation than the unbranched chains of eukaryotes and bacterial membrane lipids. The ability of archaeal lipids to resist hydrolysis and oxidation may help these organisms to withstand the extreme conditions such as high temperatures, low pH, or high salt concentrations under which some of these archaea grew. The repertoire of membrane lipids is extensive. However, these lipids possess a critical common structure theme. Membrane lipids are amphithetic molecules, that is, they contain both a hydrophilic and a hydrophobic moiety. So let us look at a model of phosphoglyceride. Well, its overall shape is roughly rectangular. The two hydrophobic fatty acid chains are about parallel to each other, whereas the hydrophilic phosphorichrome moiety points to the opposite direction. So the sphygmomyelin has a similar configuration as does the archaea lipid disulfide. So therefore, the following shorthand has been adopted to represent these membrane lipids. The hydrophilic unit, also called the polar head group, is represented by a circle, and hydrocarbon tails are deciphered by straight or wave lines. Okay, so that's all for the lipids, and thank you for listening.